Over a staggering 150 million years, milk has played a significant role in the evolution of mammals. Milk production is a defining characteristic of mammals. Milk contains a specific sugar called lactose. Lactose remains relevant to our understanding of human evolution over the past 10,000 years. This sugar has exerted a powerful influence on nutrition, immunity, and ultimately our survival as a successful species. Welcome to Anthromedia. In this video, we will explore the fascinating evolution of milk lactose digestion in humans. Lactose intolerance is the inability to digest lactose, the sugar found in milk, due to reduced production of the enzyme lactase. The evolution of lactose intolerance can be traced back to the domestication of animals and the development of dairy farming. In early human history, lactase production was limited to infancy during breastfeeding. However, as humans domesticated animals and gained access to animal milk, individuals who could continue digesting lactose into adulthood had an evolutionary advantage. In most mammals, including most humans, lactase production decreases after weaning, leading to a reduced ability to digest lactose. This can result in digestive problems such as gas, cramping, and diarrhea when lactose is consumed. However, certain individuals exhibit lactase persistence, meaning they can produce lactase throughout their lives, even after weaning. Lactase persistence is caused by genetic variations. If a person has at least one copy of the lactase persistence gene variant, they can continue to produce lactase and digest lactose without experiencing intolerance symptoms. The ability to digest lactose in adulthood is influenced by specific genetic variations near the lactase gene, such as the 13910T variant, which increases lactase production. DNA methylation patterns acting like switches for genes play a role in regulating lactase gene activity. An older form of the lactase gene, the 13910C allele, accumulates more methylation over time, resulting in lower lactase production. In contrast, the lactase persistence-related gene variant 13910C remains mostly unmethylated, allowing lactase to continue being produced. The age at which lactase production decreases varies among populations. Some populations exhibit reduced lactase levels relatively late, around 8 to 12 years old, while others show signs of reduced lactase as early as 1 year old. The reasons for these variations are not fully understood, but likely involve a combination of genetic and environmental factors, including diet. Lactose is composed of glucose, and galactose is unique to milk and is synthesized in mammary glands. The presence of lactose in all groups of mammals suggests that it was present in their last common ancestor. Originally, milk secretions likely had antimicrobial properties, providing protection against microbial infections in ancestral species. Over time, these secretions gradually became more nutrient-dense. Oligosaccharides found in the milk of most mammals likely evolved earlier than lactose. They serve to enhance antimicrobial defenses and provide immune support to offsprings. Monotremes, the egg-laying mammals, marsupials, the pouched mammals, and certain eutherians exhibit higher concentration of milk oligosaccharides compared to lactose. In eutherian mammals, lactose became the dominant carbohydrate in milk as a response to changing selective pressures and the need for an easily accessible energy source. The transition from hunter-gatherer societies to settled agricultural communities during Neolithic period brought changes in diet, with a reliance on cereals and grains. Plant domestication began approximately 12,000 years ago in the southwestern Asia, followed by animal domestication. Dogs were likely the first domesticated animals, predating crop domestication. Initially, domesticated animals were primarily utilized for meat, hides, and bones, but secondary products like milk, wool, and traction were soon incorporated. Goats and sheep were domesticated around 11,000 years ago, while cattle and pigs were domesticated around 10,500 years ago. These domesticated animals spread to different regions, reaching Europe around 6,000 years ago. Dairying and milk consumption emerged in Anatolia and Levant around 9,000 years ago, as evidenced by presence of milk lipids in pottery fragments. Fermentation techniques were employed to produce yogurt, butter, and cheese, which had reduced lactose content and improved portability. Dairying practices expanded to various regions including southeastern Europe, Poland, Romania, Hungary, and Ireland. Archaeological findings such as pottery fragments, cheese-making sieves, and lipid analysis provide evidence of milk processing and cheese-making in different regions. Fresh milk residues have been discovered in beakers in Lithuania and southern England, suggesting consumption of fresh milk over 5,000 years ago. In Ireland, buried caches of butter known as bog butter have been found in peat bogs dating back thousands of years. Excavations in Xinjiang, China, revealed the earliest evidence of kefir cheese making in Eastern Asia. Sahara Africa also shows evidence of dairying and milk processing from 7,000 years, as depicted in rock art and indicated by pottery analysis. Ancient DNA analysis has provided significant insights into evolution of lactase persistence. Lactase persistence emerged around 5,000 to 4,300 years ago in different cultures during the late Neolithic period. It coincided with the migration of steppe populations related to the Yamnaya culture in Europe. The 13910T allele associated with lactase persistence likely expanded in the populations between 7,450 and 12,300 years ago, corresponding to the development of dairy farming practices around 9,000 years ago in Europe. Lactase persistence frequency varies among populations. Northern Europeans have the highest rates, 90 to 97%, while Central Europeans have intermediate rates, 44 to 84%, and Southern and Southeastern Europeans have lower rates, 11 to 64%. Lactose persistence is relatively common in some northwestern Indian populations, but rare in East and Southeast Asian populations, as well as in indigenous populations of Australasia, Oceania, and Americas. Different populations have different distributions of lactose persistence alleles. The most common allele, 13910T allele, is found at high frequencies in British, Scandinavian, and North Indian populations, as well as in some African pastoralist populations. In Africa and the Middle East, there are other alleles associated with lactose persistence. One of them is the 14010C allele found in agro-pastoralist populations in Tanzania, Kenya, and Southern Africa. Interestingly, it is also present in the hunter-gatherer populations like the South African San and Tanzanian Sandari. The 14010C allele likely originated in East Africa and spread to South Africa through gene flow. Other alleles such as 13915G and 13907G are commonly found in the Middle East, East Africa, and North Africa. These findings suggest that lactase persistence has independently evolved in different human populations, offering advantages to individuals who can digest milk. The frequency of lactase persistence has increased over time in Europe. During the Bronze Age, its frequency was around 7% in Central Europe. In the Iron Age, it reached up to 19% in certain cultures. Lactase persistence frequency varied in the Middle Ages, exhibiting heterogeneity among different populations. The evolution of lactose digestion and the prevalence of lactose persistence have been associated with potential evolutionary advantages. In the high altitude environments with limited sunshine, milk consumption prevents vitamin D deficiency and rickets, as lactose promotes the uptake of calcium. This may explain the high frequency of lactase persistence in northern European populations compared to those consuming milk primarily as cheese in the Mediterranean. In arid environments, the water content of fresh milk increases survival chances for lactose digesting individuals, specifically among desert dwelling pastoralists. Diarrhea and water depletion in lactose intolerant individuals may lead to selective disadvantage. This is supported by the high frequencies of lactose persistence in hot areas such as Middle East and North Africa. Cultural, nutritional, and environmental factors, including preferences for low lactose dairy products, availability of alternative foods, seasonal influences, mobility, and livestock practices, have also influenced lactase persistence in certain populations. Genetic factors and associations between lactose fermentation in the colon and energy production from short chain fatty acids have been observed, benefiting individuals who cannot digest lactose well. While lactose's significance throughout mammalian and human evolution is profound, there is ongoing research to fully understand its implications and potential benefits beyond milk digestion. Milk continues to captivate researchers, shedding light on our adaptive flexibility and response to various challenges. Its story is an ever unfolding tale of evolutionary intrigue.